Hi, I'm Sarah Taylor. I'm the collections manager for paleobotany and for the herbarium here in the Biodiversity Research Collections. So currently we are in the dry collections room where we have collections of invertebrates, plant specimens, paleobotany, and vertebrates and parasites. And so today I'll talk to you a little bit about the herbarium and the paleobotany sections. So we store our herbarium specimens flat in folders. And a lot of people have never heard of a herbarium before, so let's start with the basics. A herbarium is a collection of dried, pressed plants that are mounted to paper with information about the plants written on the paper. And a herbarium can range in size from a few dozen specimens in a personal collection up to millions and millions of specimens in the world's largest institutional herbaria, which are in Paris and in London. Okay, so this is a specimen that was collected by Henry Ballou in 1898, and he was the founder of the first herbarium here when this was still called Storrs Agricultural College. And he founded the herbarium with student collections. The first herbarium that we had was created from about 120 student collections that were made in the fall of 1897. And this is a specimen that he collected here on campus. It's yarrow, which is a common plant that you'll see around campus today. So our herbarium started out very small with about 120 specimens, and we now have about 220,000 specimens. There is a lot of work that goes into collecting and documenting the plants that we have in our collection. So first, there is the work that it takes to actually go out in the field and collect plants. So we collect our plants, we write down notes in our field notebooks about where and when we collected the plant. We take notes on color, what other plants were growing nearby, and then we have to bring them back into the collection and press them and mount them on the paper in order to make a scientifically useful specimen. So this is a herbarium specimen of Jack in the Pulpit, which is native to Connecticut. And there's a lot of data that we can get from this one specimen. So for example, we can see whether the specimen is flowering or fruiting. We can look at the leaf shape. We can see both the top and the underside of the leaf. And the spathe has been cut away so that we can see the female flowers down at the bottom. We also can figure out exactly where and when this specimen was collected, which helps us place this plant in space and in time. It also tells us a bit about the habitat. It was found in a mesic trailside in the woods. And it also, importantly, tells us the color of the plant. So sometimes color is really important, especially of flowers, but it's a property that fades over time in herbarium specimens. So if you have a specimen that's 100 years old and it doesn't say what the color is, we have no way of figuring that out currently. We also, when we're collecting these specimens, don't know what they might be used for in the future. For example, we can isolate DNA from herbarium specimens. And in fact, we can isolate DNA from specimens that were collected before we understood that DNA is genetic material. So when those plants were collected, the botanists had no idea that someday someone would be able to sequence their genetic material. And in that way, we also know that the plants that we collect today will probably be used to answer research questions that we haven't even imagined yet. So we have to preserve them to the best of our ability. Some plants don't take particularly well to being pressed. They would get ruined if we actually pressed them flat. And that goes for our bryophytes, which are mosses and hornworts and liverworts, and also for lichens. And so for those plants, we dry them in paper lunch sacks, and then that preserves their three-dimensional structure. And then they get stored in these archival paper folders, which protects them. Okay, so now that we have talked about how herbarium specimens are made and the effort that goes into them and how they're filed, let's actually go and talk about how we use the data that we've collected from these plants. Okay, so here we have a specimen, a herbarium specimen of a species of rose that's native to Connecticut. It's called Rosa palustris. And you can see we have these folders here, and they're full of over 80 collections of Rosa palustris from across Connecticut. And the earliest ones are from about 1815, 1810, up to 2018. And having specimens of one species through a time course allows us to address several different kinds of research questions. One of the main topics that herbarium specimens are currently being used for is to investigate climate change. So of course, for climate change, we want to look at changes over time, and for this, herbarium specimens are perfect. So we can look at what the earliest flowering date of these plants has been over time. And that is called phenology. Phenology is the study of the timing of life cycle events in animals and in plants, for example, flowering or fruiting. So when we take a look at the flowering times for Rosa palustris over the last 100 years, we might discover that the timing of flowering has become a little bit earlier over time. And in fact, when I looked at all of these, that is what I found when I plotted the data, that Rosa palustris is now flowering a couple weeks earlier than it was 100 years ago. So there are several other kinds of research that we also can use these herbarium specimens for. So for example, scientists have used herbarium sheets to investigate plant and insect interactions by looking at leaf damage on herbarium specimens that was caused by insects. We can also use herbarium data to target where to search for other organisms out in the field. For example, there was a study that was done where researchers wanted to know where they could go to find a rare butterfly, and they had been searching without very much success. But they looked then at the herbarium data on labels for the plants that they knew the butterflies eat. And so they could then target those locations to go and find the rare butterflies that they were looking for. We can also use herbarium specimens to create a floristic inventory of a geographic area. So for example, we have a 30,000 specimen gift of specimens that document the floristic diversity of Worcester County in Massachusetts. We also have been assisting with a project that is documenting the floristic diversity of Concord, Massachusetts. And the interesting thing about Concord, Massachusetts is that they have created floristic inventories 5 times. And Henry David Thoreau created the second inventory. And what we're now finding out is that since the time that Henry David Thoreau created his inventory, the diversity of Concord, Massachusetts has actually declined by about 25% in terms of the plants that are there. We can also use herbarium specimens to track the movement and migration of invasive species over time, and we can use them to monitor populations of rare plants. So those are just a few of the research applications of herbarium data. Okay, so we've talked a lot about herbarium specimens and how we use that data, but I'm also the paleobotany collections manager. And so those are plant fossils, and this is my favorite plant fossil. It's about 320 million years old, and it is the trunk of a plant called Lepidocarpon. And you can see all of the little leaf scars that came off of the trunk. And this is a different kind of a tree. It's a little bit like tree ferns. It's related to tree ferns, and it's called a tree lycopod. And 320 million years ago, northeastern North America was really different. It was really hot and swampy, and there were no flowers because flowers hadn't evolved yet. There were no oak trees and no grasses. But these tree lycopod forests were really vast. There also were giant insects, such as dragonflies the size of crows. There were enormous amphibians. We think partly that's because the concentration of oxygen in the atmosphere was far higher than it is today. But then, around 299 million years ago, the climate began to change. It cooled, it got dry, and these gigantic lycopod forests went extinct. At the time, there were fungi, but they had not yet evolved the enzymes that could decompose the wood in these tree trunks. So instead of rotting, all of the tree lycopods sank into the swamps and eventually, over millions of years ago, were turned into coal. So these fossils are a good opportunity for us to study extinct plants. These are not around anymore. However, even right by campus, you can see some of their relatives that are alive today. So this is a plant called Dendrolycopodium obscurum. It's a little tiny club moss, and it is related to the giant tree lycopods that went extinct so long ago. And if you were able to look really closely, you could see the tiny little leaves that have almost the same pattern as the leaves on the tree trunk. And this plant was just collected right behind Hillel House. So we can use our paleobotany collection to study extinct organisms, and we can also use those fossils to compare them to the relatives that are alive today. So I hope you've enjoyed hearing about the herbarium, and we hope you get a chance to come visit soon. Thanks.